Hi everyone, thank you for joining my presentation. My name is Be² Silva and I'm a PhD student in the Department of Chemistry and Biochemistry at UNLV. The topic of my presentation is a study of beryllium ion interaction with magnesium-independent carboxylate-rich model peptides. To place my study in terms of the bigger picture, my dissertation research is part of one of the steps involved in the drug development process. The drug development process includes several steps. The first step is basic research. This is where the best targets for treating or preventing a disease are identified. And these targets are usually proteins associated with a particular disease. And scientists would explore ways to interfere with the activity of this target protein. The second step is drug discovery and development. This is where lead compounds for potential drugs are identified and screened. My dissertation study belongs to this step. Once a successful drug candidate is discovered, it must be further studied through preclinical and clinical research before it can become an approved drug. So in my presentation, I would introduce our target protein and its significance, go over background and basic research that helped us to derive the hypothesis for our study, explain the approach we're following in order to discover a lead compound, and then go over some experimental results and finish with conclusions and future directions. The target protein we're interested in is called Glycogen Synthase Kinase 3 beta. It's abbreviated as GSK3 beta. As a kinase, its function is to activate or deactivate a downstream target protein by adding on a phosphate group. This process is called as phosphorylation. Tau is one of the proteins phosphorylated by GSK3 beta. However, hyperphosphorylation of tau by GSK3 beta can cause tau protein to form tangles inside brain cells. Tau tangles are one of the hallmark pathologies of Alzheimer's disease, and hyperphosphorylation of tau by GSK3 beta accelerates the accumulation of beta amyloid plaques in the brain, which is the other hallmark pathology of Alzheimer's disease. Because of this, GSK3 beta is a potential target for developing drugs to slow down the progression of Alzheimer's disease. However, there are some challenges to developing drugs targeting GSK3 beta activity. The ATP binding domain in this kinase is quite conserved among other kinases in the kinase family. And also, GSK3 beta protein is involved in many most cellular pathways. So, we're interested in discovering drug molecules that can selectively target GSK3 beta with minimal influence to its activity in other cellular pathways. We believe beryllium ion is a promising candidate to help discover potent and selective inhibitors for GSK3 beta, and our hypothesis is based on previous data published from our lab. Before beryllium, lithium has been used as a classical inhibitor of GSK3 beta in research studies, and it's also used as a treatment for bipolar disorder. Studies done in our research group reported that beryllium ion, which is actually neighboring to lithium in the periodic table, is actually 1,000-fold more potent than lithium in inhibiting the GSK3 beta activity. Most GSK3 beta inhibitors, including lithium, inhibit GSK3 beta activity by phosphorylating a serine residue at the position 9. However, surprisingly, beryllium-mediated inhibition of GSK3 beta did not follow by phosphorylation of serine 9. And in another study from our research group showed that lithium did and beryllium did not introduce changes to one of its downstream target proteins called beta-catenin. This evidence suggested that beryllium must be inhibiting GSK3-beta activity by binding to the protein in a mechanism different to that of lithium, and also has some selectivity on its effects on the other cellular pathways. Beryllium is a positively charged ion, and it's in the same group as magnesium in the periodic table. Also, GSK3-beta is a magnesium-dependent kinase, and it's been discovered that magnesium binds to the amino acid residues aspartic acid 200 and 264. Aspartic acid is one of the negatively charged amino acids, while the other one is glutamic acid. They're negatively charged because their carboxylic acid groups are predominantly deprotonated at physiological pH. In this ball and stick structure of GSK3-beta protein, the negatively charged amino acids are shown in red color. There appears to be multiple sites that can work as a negatively charged binding pocket because there are a number of instances in the structure where aspartic acid residues and glutamic acid residues are in a close proximity. Since magnesium binds to aspartic acid, we can expect beryllium is able to bind similar binding sites and may also be able to bind to sites that cannot be reached by magnesium because beryllium has a smaller ionic radius than magnesium. So for our study, we decided to use some model peptides featuring aspartic acid and glutamic acid mimicking small binding pockets and compare the binding of that against beryllium and magnesium. We expected to find binding interactions specific to beryllium and not for magnesium. To study the binding between beryllium and magnesium with the model peptides, we used isothermal titration calorimetry. This is one of the best methods available to directly measure binding parameters for a given binding interaction. We can inject our ligand, which in our case is beryllium or magnesium, through an automated syringe into a sample containing the peptides, and the temperature of the sample cell is maintained same as the temperature of the reference cell. Depending on the manner of binding, heat is either released or absorbed, and the instrument would supply less or more heat to compensate for that temperature change, and this is measured in terms of differential power. This heat change per injection can then be integrated to obtain enthalpy change per molar ratio between ligand and cell, giving us a binding curve. The binding curve directly gives us the enthalpy, the binding affinity, and the number of binding sites. And using these data and the temperature, we can calculate the Gibbs free energy for the binding and the entropy, delta S. We compared the binding of beryllium and magnesium ions to two simple peptides, one made with 4 aspartic acid residues and one made with 4 glutamic acid residues. Both have 4 carboxylate groups each, and all the experiments were performed under the same buffer conditions. Beryllium displayed a stronger affinity for the 4D peptide and a weaker affinity for the 4E peptide, and magnesium didn't display binding with either peptide. This again provided us evidence that beryllium has a stronger affinity towards carboxylate groups compared to magnesium. Another important observation was the carboxylate groups in the 4D peptide have a restricted conformation compared to the carboxylate groups in 4E, which has more freedom of rotation. So we think the weak affinity of beryllium towards the 4E peptide can be attributed to having an entropy cost to bring the carboxylate groups to form a small binding pocket. We performed binding studies under the same experimental conditions to compare the binding of beryllium to peptides with a decreasing number of aspartic acid residues. We did this by replacing an aspartic acid residue with a neutral glycine residue. The peptide made only of glycine residues was used as a control, and since it's a neutral peptide, it should show no binding. We were able to see a decrease in binding affinity with the decrease of the number of aspartic acid residues, and the stoichiometry of binding between beryllium ion and the peptides was 2 for the 4D peptide, 1 for GDD peptide, 1 for GDGD peptide, and none for the GGDD peptide. This indicated that beryllium binds to small binding pockets with aspartic acid residues that are alternatively spaced. In conclusion, we observed that unlike magnesium, beryllium can bind to small binding pockets featuring aspartic or glutamic acid residues with a higher affinity for aspartic acid. The titration with beryllium with the peptides it bound to were all thermodynamically favored, and these thermodynamic data can be used to design lead compounds targeting GSK3-beta. A limitation of our study was conducting these titrations at pH 6 to 5.8 due to the poor solubility of beryllium at pH 7 aqueous buffers. For our future studies, we intend to follow these titrations at a near physiological pH as well as follow ITC titrations between beryllium and the full-length GSK3-beta protein. I would like to acknowledge our funding sources and many thanks to my advisor Dr. Ronald Gary and the members of Gary Lab. Again, thank you for joining my presentation.